briefly introduce our speaker, Dr. Chalasani. He's an associated professor since 2016 in the Molecular Neurobiology Laboratory, the Salk Institute for Biological Studies. And Dr. Chalasani did his PhD at the University of Pennsylvania in the Department of Biology. And the field of study was neuroscience, and the advisor was Jonathan Raper. And then he moved to his postdoctoral research at the University of California in San Francisco and the Rockefeller University. And that was also in neuroscience with the advisor Corey Barman. And he also received many honors. So I would like to invite Chalasani to start his talk, and I really look forward to hear about his work.
Okay, thank you, Rita. Thank you, organizing committee. Can you guys all hear me? Yes? No? Yes, perfect. Thank you, everyone. So I am really excited about, thanks for the opportunity to present our work today. I'm excited about this topic. This is a new area for us. We've been thinking about how different tissues are communicating with each other. And we think that there is a mesoscale level of organization in C. elegans and perhaps in other animals, where individual cells cooperate to be a tissue, but then the tissue itself is able to talk to one another in affecting physiology and behavior. So today, what I'm going to do is tell you about our recent study and a little bit of unpublished data to talk about how we think this project or this particular pathway is working. So let's get started. So before I tell you the research, I'm going to introduce you to the people who did all the work. So the project was started by Eunice La, a graduate student. After finishing up her PhD, she now is a patent officer, and I think the chief patent officer for 10x Genomics. She lives in St. Louis. But this project was really completed by a group of people, in particular, Molly Matty. So Molly is an assistant professor at University of Portland today. Jess Haley, a grad student, a postdoc, Anupama, and our research scientist, Kirti. So a lot of people have worked on this project and are continuing to pursue asking this question about how an internal state is changing behavior. Okay, so here's the plan for today. I'm going to give you a brief introduction of what is the question, how do we think about this question. I'm going to show you data which is mostly published earlier this year, that food deprivation, that is removing an animal from food, affects its behavior. And then I'm going to show you two other topics. One is what is happening in the neurons that are sensing these signals. And then finally, I'm going to tell you about a social communication between well-fed animals and food-deprived animals and how that affects behavior. Okay, so let's get started. I know at least I like cartoons a lot, so I'm going to start off showing you a little cartoon. One of my favorite characters is Mr. Bean. He's a comedian in the UK, and there is a lot of different things that he has done. And this is one of those commercial things that he has shown. Unfortunately, you may not be able to hear anything, but you can watch this at your own pace if you like. So when we are hungry, things look really hard. Our training doesn't matter. Our skills don't matter. Hunger has a huge effect on our responses, right? Obviously, we get caught when we are hungry, but the point I'm trying to make here with this is to tell you, and I think we all appreciate that, is if you didn't have lunch today, you're not going to be in a good mood. Your behavior is going to be different. Your response to stimulus is going to be different. So this is the larger question. How does hunger modify animal behavior? The problem with this statement is really with the word hunger. Now, we can ask a human to tell us if they are hungry or not, but if you want to study this in an animal, you cannot ask the animal whether the animal is hungry. Sometimes you might be able to decipher that the animal is hungry based on other assays, but what we are really doing in these experiments is we're causing food deprivation. We're depriving them of food for a certain amount of time. So that's the behavioral modification, or that's the change that we are doing. And we're asking, how does food deprivation modify animal behavior? Okay. So this is the question that we are trying to address today, uh, for the next 50 minutes or so, roughly. Now, it has been appreciated for decades now that animal physiology changes with food deprivation, right? And nutritional status is going to affect physiology. Now, we know that food is absorbed in the intestine. The amount of food that we're absorbing is then sent off to the brain. The brain then shuts off our ability to eat more. This energy is being stored in fat stores and in other deposit areas. This energy is utilized when we do action, when we move, for other activities in our body. Uh, and then this energy is also converted back into storage. So this cycle allows us to regulate the amount of food we are taking with the amount of energy that we are expending during the day. Now, what has not been appreciated as much, at least in my opinion, is the effect of the change in nutritional status on behavior. While physiology has been well studied, behavior has been harder to study. Now, in the context of the human and the mammalian brain, there's a little bit known about it, and most of it comes from studies of this particular sensory system called interoception. So, interoception is the idea that there is a sensory system that is sensing internal state. And this sensory system includes a number of sensors. They are mechanosensors, thermosensors, and chemosensors. And they are relaying information from the body organs to the brain. And they are relaying typically food status, they're relaying arousal status, and they're allowing us to detect things inside our body. Now, this system has been studied to some extent in both rodents and in mammals and in humans, mostly from fMRI studies or from stroke patients or epilepsy patients. And what we find is that there are different areas in cortex, particularly anterior cingulate cortex or insular cortex, which are playing a critical role in affecting our internal status. Right? So the sense of internal organs is basically interoception. This is sensing hunger, heart rate, thirst, etc. The insular cortex, basically right insular cortex, is where you are processing feeling of well-being. It's an emotional center. Mid-cingulate cortex is thought to be about environmental postures, environmental temperatures, and body postures. And these are parts of the brain that seem to be affected in sensory disorders, like what we see in children who have been diagnosed with autism or schizophrenia or any of these conditions. Now, while this kind of work is interesting and exciting, we really don't have a mechanistic understanding of what's happening. How do you know that a person is hungry? How do you know an individual or an animal is hungry? What is that signal that tells you that you are hungry? What is the signal that communicates that from one organ to another? And what is the change in the nervous system that you're going to get at? Now, is it true that food deprivation changes behavior? And food deprivation changes behavior, we know from our personal experience in humans, but does it also happen in other animals, right? And that's true. More than a decade ago, Jing Wang's lab at UCSD showed that, at least in the fly, there's a big difference between an animal that is well fed and an animal that is starved. In their idea, starvation is 6 to 10 hours. away from food. Okay, so here's the experiment. This is a picture of a fly that I got out of Wikipedia. I'm putting them in a plate. There is this animal is now searching for food in this arena. If it's a fed animal, it spends a lot of time exploring the plate. If it's a starved animal, it doesn't move as much. And so they can tell some animal is fed or starved. So starvation is changing the rate at which this animal is moving, or the amount of space that this animal occupies or searches. There was a true just in a fly. It turns out that's not the case. About 7 years ago, Thomas Horvath at Yale showed that AGRP neurons in the hypothalamus are critical for playing food intake. They're also affecting the animal's ability to eat, but they have an additional role, and they have an additional role in affecting behavior. So clearly, a group of neurons that are regulating how much the animal is eating is also affecting behavior from the animal. So again, implying that there's a link between food deprivation and behavior that the animal can execute. Now, over the last decade, I think this part of the physiology of the interorgan communication has become really well studied, or people are really getting excited about it, which is this idea that the gut and the brain are talking to each other. So the gut-brain communication is considered the most important thing in regulating mood, in regulating behavior, even in affecting many conditions like depression, anxiety, maybe even larger conditions. More importantly, the gut, as we know, is colonized by a microbiome. And this microbiome plays a really critical role in affecting the mood and our well being and our behavior. So there's a lot being studied about this, but I would argue that despite all these studies, we are still unable to get a handle on the precise machinery that is regulating each of these steps. Again, I reiterate, how do you know that an animal is hungry? What is the signal that goes from the animal, from the organ that is sensing that hunger state or the food deprived state to the brain? And what are the changes that you can occur in a brain, right? So if you want to study something like this, I think using a simple system is the best place to go after a question as complex as this. And I can't think of a simpler system than the nematode C. elegans. So this is a picture of a C. elegans. This is the DIC image up on the top. This is the head of the animal, the tail of the animal. It has now laid 2 eggs. The eggs are here in the gonad. It has muscles on its body that allow the animal to move, and it has neurons, and it has intestinal cells, right? It also has a few other tissues, like it has a pharynx, it has a hypodermal cell that is underneath the cuticle. And you might have heard of C. elegans before, given all of the presenters who have presented in this series, but this is a really simple animal. And here's kind of an outline of what this animal has, right? It has precisely 959 somatic cells. I'm not talking about germ cells in the gonad, this is just somatic cells. It has just 20 cells in the intestine. It has 95 body wall muscle cells, and it has 302 neurons. Given this small number, it's easy for us to understand what is happening at the level of individual cells and at the level of individual tissues. So we have an ability to probe each of these cells or each of these tissues for function, either by behavior assay or by reporters, because the animal is completely transparent. You can also change any of these cells or any of these tissues. You can stop a tissue from sending signals, from receiving signals. You can using genetic tricks, and you can make transgenic animals in about 4 days, basically. So this is a really simple system for studying questions like this. Okay, so now you have a system, but does this system change its behavior when you don't have food? Like what is known about this guy? So here is an example of that. So this is a very simple experiment. So imagine I'm going in, these animals eat bacteria and they live on agar plates. What if you were able to, they live on agar plates and they eat bacteria. So what if you came in You took this animal that was sitting on food and you moved it to a plate that doesn't have any food. Now, the animal doesn't know what you did, that you were involved in moving them. We think the animal is probably feeling that it made a mistake. It was sitting on food, happy, made a mistake. Suddenly, there's no food. So, what it does, it looks for food and it performs this interesting behavior that we can see. So, when the animal is sitting on food, it moves very slowly, keeps eating, makes very few turns. When you move it away from food to a plate without any food, it's going to search that local space for about 15 minutes. After 15 minutes, when it can't find any food, it leaves that local space and goes off and searches the broader area. If you change the quality of the food here, you change the duration of search. If you change the size of the food here, you change the size of the area the animal is searching. So clearly, if you deprive the animal from food, you're able to change its behavior. You go from moving slowly to moving a little bit faster, to turning, to finding, to searching. The longer you food deprive them, the more behavior change you get. Now, if you go more than 30 minutes, you're going to go straight. Mostly, you're not going to turn as many times. This 15 minutes also seems to be important for affecting attraction to salt. So if you take animals, you starve them for 15 minutes, I should say, sorry, you food deprive them for 15 minutes, you also change their behavior to salt. I mean, animals are normally attracted to salt, but if they are in a buffer without salt, now they're repelled away from salt. Okay, so we know that something is happening around 15 minutes. But what about longer amounts of food deprivation, right? So you start off with animals on food, you move them to a plate without any food for maybe 0 to 6 hours, and then you ask them to find a spot of a volatile chemical that they love. And this is in a chemotaxis assay. So when you do control animals, you start here, you put them in the plate, you get pretty nice behavior. If you do 1 hour of food deprivation, the behavior seems to match somewhat. 2 hours, maybe similar. But if you start them for 6 hours, now things become very different. So it seems like between 0 to 6 hours, if you food deprive C. elegans, animals have pretty normal looking locomotion, but they might have some changes in behavior. After 6 hours, they seem to go into a different state. So we started out our project thinking, what's happening? How does the animal know what's happening between 0 and 6 hours? What are the changes there? We didn't want to go into what happens after 6 hours because we think that after that, we might have breakdown of fat deposits, which we can identify. And so we might be affecting multiple things in the animal physiology. So we're looking at an early step, an early sensor of food deprivation. At this stage, the animal is not changed a lot of its physiology, but is still able to alter its behavior so that it can continue to go back and get into a controlled state where it's back on food and not continue to food deprive and become starved later on. Okay. So this is kind of where we started out our project, and this is kind of where we were headed. So here is our behavioral experiment. We grow up animals to adults on bacteria. We move them either to a plate without food for 3 hours or to a plate with another bacterial on, which are our controls. And then we test them in a behavior assay. And the assay that we are using is a little bit of a complex assay. The assay has a barrier in the middle. The barrier is made of copper. Copper is a repellent. It's a toxin. When it gets into the animal, it can poison enzymes, and it can damage the animal's physiology and system. You put a spot of something they like on one side of the barrier, you stick animals on the other side, and then you ask how many of them will cross this barrier over a period of time. Okay, so that's the behavior that we are using to measure in this experiment. Okay, So I'm going to show you a video. This is a zoomed out video. You're going to see C. elegans as little squiggles. The barrier is right down here. This is a spot of something they love. And we're asking how many of these guys get across, right? So on the left is well-fed animals. On the right are 3 hours of food-deprived animals, okay? So let's see if this thing will play. So I tried to click them as quickly as I could, but what you're seeing is little stuff buzzing around. Very few animals are crossing in this side, and then you see more animals are crossing this barrier on this video on the right. So it looks like if you're well fed, you seem to avoid this barrier. If you're food deprived, you don't seem to be avoiding the barrier. You're getting across and getting to the food. Now, ethologically, that kind of makes sense. I mean, if the barrier represents risk, if you're well fed, you don't need to take as many risks in life. But if you're food deprived, you might want to take that extra risk because you don't want to continue being food deprived. You want to try and go back to being on food. So ethologically, it kind of makes sense. But what about mechanistically? What is the mechanism for something like this happening? What is this behavior change that we are talking about? Okay, so when we quantify, this is quantified by an index. This is basically the number of animals that cross the barrier or the total number of animals in the assay. If you're well fed, maybe about 40% cross the barrier. If you're food deprived, about 80% cross the barrier. So you have twice as many crossing the barrier if you're food deprived. So that gives you a basic phenotype that now we can start to ask what is the underlying machinery that is driving this behavior change. Okay. So it turns out we also confirm to make sure that if you deprive them from food for 3 hours, you're not affecting fat stores. Now, you could come back and ask me, Shrek, this sounds exciting, but how do you know that you have not changed other aspects of animal physiology? We don't. 3 hours of food deprivation is still food deprivation. We might have changed things here that we cannot identify yet. All we can do is measure how much fat this animal has. And it looks like 3 hours of food deprivation does not have a change in fat. Okay. But there might be other changes that we have not been able to detect yet. Okay. Now, what is this behavior? We find that the behavior is transient and it's reversible. So if you start off this behavior with well-fed animals and you ask, well, what happens if you start them for different durations of time? You start seeing the response at about 2 hours, where it's now significant. And then at about 3 hours, the response seems to be mostly maximal. But longer food deprivation doesn't seem to affect as much. So 3 hours and 6 hours, not much of a change going on. Now, if I take animals that are 3 hours of food deprived, put them back on food for different durations of time. If I put them back on food for 1 hour, I still maintain the difference between the food deprived and well-fed animals. However, if I put them back on food for 3 hours before testing them a second time, now the difference goes away. So it looks like what we have done with this 3 hours of food deprivation is you have changed behavior, you have made them cross that barrier, but if you put them back on food, they regain that sensitivity to the barrier. You haven't done something permanent. This is a transient change and it is reversible change. So it allows us to now start to ask about these transient reversible changes in the system. Okay. Now, so because there are two things in this experiment, right? On the one side, you have something they like. In this case, we're using popcorn butter, which is diacetyl. The barrier is made of copper. So you have more animals crossing the copper barrier. So what you could have done is when you're food deprived, you might become more attracted to the odor, or you might become less repelled from the repellent. So which is it? And you can do that by testing them independently. So it turns out if you look at animals that are well fed and you just measure the response to copper, more animals cross the barrier, saying that now this is a reduction in response to the copper, but the response to the odor is not affected. So it looks like what you're doing in C. elegans is when you food deprive them, you're reducing their sensitivity to copper, but not affecting their sensitivity to odor. Now, this is different than what's being shown in flies, where food depriving Drosophila seems to increase their attraction to odorants without affecting repellents. So it could be that different species have different responses to food deprivation. It also might represent something about their natural ecology. So if you are a fly and you're able to fly, you might be able to cover large territory. So perhaps ethologically speaking, behaviorally speaking, you might be better off increasing your sensitivity in your attractant neurons. And if you're an animal that is living in soil, eating bacteria, maybe what you want to do is change your sensitivity to repellent. It's pure speculation. I don't have any data because I don't understand yet, or we don't understand yet, what's happening in the natural ecology at the moment. Okay, so we know that you're reducing copper avoidance. Now, is this phenotype specific to copper, or does it also affect other repellents that they see that the animal can detect? So we tested different repellents, and we found that most of the time other repellents are not affected. So we tested quinine, which is from the bark of a tree. It's a strong repellent for C. elegans. We tested high concentration of sodium chloride, which is an osmotic shock. We tested fructose, which is also an osmotic shock, and none of these things are affected. There might be a small effect with an intermediate concentration of fructose, which is still significant statistically. But I think that broadly speaking, this is not a broad change in repellent sensitivity. What you have done is change sensitivity to copper, but not to other things. Now, why this is the case is still unknown. It's not clear to me that copper is found more readily in the environment, that the animal needs to have a particular pathway for copper. But this is the observation that we made. Now, this copper sensitivity is affected, but there is a threshold. If you keep adding more and more copper, at some point the behavior starts to disappear. So now you're adding 200 millimolar copper, which is 10 times what we typically do in our experiments. At that point, there's no difference between a well-fed and a food-deprived animal. So it says, yeah, you're changing sensitivity of copper, but there is a range. After that range, the sensitivity kind of goes away. Okay, so this is kind of where we started out thinking about the next steps in this behavior. So, we are a behavior lab, we're a neuroscience lab, so we have sophisticated tracking machinery. We track these animals. I showed you a video earlier, but this is now shown by tracking data. It looks like well-fed animals mostly stay away from the barrier, whereas food-deprived animals are getting ready, are getting close to the barrier, and the barrier is shown down here. Time is in the bottom. So you're starting out in the blues and you're ending up with reds. And so more time means that more animals are crossing the barrier in the food deprived state. So kind of confirming what we had observed in the videos and all of the experiments that we had done before. So what is this question? What is food? So it turns out food is a really complex stimulus for C. elegans. Food includes mechanical nature. So C. elegans is crawling in food, so the touch of food. It also includes volatile signals because food can release chemicals. And now you can ask if those chemicals are affecting the behavior. So the question then when we started to ask is, what aspect of food is really affecting this response, right? So here is that question. So if the chemical signal from food is important, then if you add that signal just to the lid, you should be able to rescue these food deprived animals. Right? Is that true? So we did this experiment. We take well-fed and food deprived animals. We stick the odor of food on the lid, and we ask, when you have odor of food on the lid, you still see a difference between well-fed and food deprived, which means it is not the chemical signal. It is also not the mechanical signal, but it's the absence of food inside the animal that is really changing this behavior. And so this is where our project took a different turn, kind of an unexpected turn, because Eunice and later Molly started to ask, what was this signal? How did the animal know that there was no food in its belly or in its intestines? So this is kind of what I'm going to show you for the next 10 minutes, that the signal is likely from the intestine. It is dependent on starvation, it's dependent on food deprivation. This is tunable and it's reversible. And the signal is going to talk to neurons to change the behavior. Okay. So what is this signal? So in order to get a handle on the signal, we started to think about global signals that are required or at least work in sensing food. So here is one of those pathways. We've all heard of glycolysis pathway. Glycolysis starts off with glucose. You add a phosphate group to the sixth carbon. You break it down, and then this is now entering into Krebs cycle to start to make ATP and other products for cell survival, right? So there is a really interesting bit that this particular molecule does, glucose 6-phosphate. Glucose 6-phosphate can enter a cell and bind a family of BHLH transcription factors. This is a conserved family called MONDO. MONDO binds this protein, interacts with its binding partner MLX, goes into the nucleus, and then turns on transcription from a variety of genes that are all dependent on glucose levels. So what is happening here? When you have glucose in the cell, MONDO takes that glucose 6-phosphate, takes it into the nucleus. When you don't have glucose 6-phosphate in the cell, MONDO is sitting in the cytoplasm. So our model is that MONDO in the cytoplasm means the animal is food deprived, because MONDO in the nucleus means the animal is well fed since you have glucose, right? So here is a metabolic signal that's telling you what the internal state is. Now, is that true? Is that the right model? So we tested mutants in C. elegans, in MONDO and its binding partner MXL. It turns out in C. elegans, these mutants are viable. There are very few species where mutants in MONDO are really viable. Yeast is one of them, C. elegans is another. In mammals, MONDO mutants are not viable, so you can't really do this experiment. You might be able to do a conditional knockout of MONDO in specific cells. And that's something that could be interesting to test out, particularly if there is a lab here thinking about mammalian signaling pathways. So we then asked, okay, so we knew that MONDO was defective. Where is MONDO acting? So in C. elegans, we have a little trick. We can take a full-length copy of the cDNA, express it in different tissues, and ask what is able to rescue the mutant phenotype. So if MONDO was functioning in the intestine, expressing the full-length copy under an intestine-specific promoter should give you normal behavior. If MONDO was in body wall muscle, you would get the same thing. So we tested all neurons, body wall muscles, and intestine. And it turns out MONDO seems to mostly function in the intestine. There is some effect by expressing it in body wall muscles, but majority of the effect seems to be in the intestine. Doesn't seem to function in the neurons. So this would argue that the molecule that is likely sensing food deprivation is in the intestine. And the intestine is where MONDO seems to function. This is a transcription factor. It binds glucose 6-phosphate and kind of fits in with our overall model of MONDO playing a role in sensing food. Now, if that was true, then you should be able to see that effect, right? So here is that effect. So this is a translational fusion. Attach GFP to the protein. When you do that in well-fed animals, MONDO is the nucleus, as we would expect. When you food deprive them for 3 hours, MONDO comes out of the nucleus and goes into the cytoplasm. You don't see this nice intestinal nuclei. Instead, you see diffuse staining in the intestine. Okay, so that's good. At least fits with our model. But then what's special about 3 hours? Why 3 hours of food deprivation? What is so unique about this timeline? It turns out you start off on food most of the signal seems to be in the nucleus. You start to food deprive them. At about 3 hours, most of the signal now stands to be in the cytoplasm. So we think the reason 3 hours is important is because it takes about 3 hours for the transcription factor to get out of the nucleus and into the cytoplasm. So there is some kind of regulatory machinery that's keeping the transcription factor in the nucleus, even though the animal is still food deprived. This might have to do with the kinetics of the on and off rates of how this transcription factor translocates to this nucleus and comes out of the nucleus into the cytoplasm. So those mechanistic details are unknown at the moment, but the 3 hours gives you this timeline of what is important, of why the 3 hours are important for food deprived animals to change their behavior. So when Mondo is in the cytoplasm, what does it actually do? That has been unknown so far. We know what Mondo can do in the nucleus because it's a transcription factor. It causes transcription of different genes. But what does it do in the cytoplasm? And that's an unknown question. And that's where I think we started to think about what is its next step or what is its binding partner for Mondo. It turns out Mondo interacts with a second transcription factor called HLH30 in C. elegans. And this is PTFB, which is the major transcription factor regulating autophagy in mammalian cells and in all cells that we know of. So given that Mondo is interacting with HLH30 or PTFB, we think that PTFB is perhaps also involved in this pathway. So we tested PTFB, and this is kind of what we use the same strategy of using a translational fusion where we attach GFP to PTFB and we ask what's happening. And there's a previous publication that showed that the PTFB binds EBOX proteins, at least proteins that have EBOXes in their transcription and their promoter elements, and turn on transcription. When PTFB goes into the nucleus, it shows up in intestinal nuclei. That is when the transcription factor is active. When PTFB is inactive is when it's and it's translocating back into the cytoplasm when it's inactive. So similar to Mondo, PTFB can also get out of the nucleus and sit in the cytoplasm when it's not active, right? Okay, so what happens? We measure this over time, and it looks like the localization of PTFB in C. elegans intestines is dynamic. So you get rapid changes. It goes into the nucleus, comes out of nucleus over a period of an hour or two. It doesn't seem to fit the 3-hour thing that Mondo does. So the regulation of Mondo and PTFB in intestinal cells as a consequence of food deprivation is still confusing to me and confusing to our group, right? But when you take a mutant, it is not responding to food deprivation. So clearly this gene is required for animals to alter their behavior when they are food deprived. So this gene is required, the localization is dynamic. So let me actually show you a little video to demonstrate what I'm talking about. So we have the same animal that is stuck on an agarose pad, and we are imaging it over a period of time, using a spinning disc. And you're looking at a video of these guys. So animal starts off at the nucleus and then starts to go back out. And then now you start to see the nuclei coming back in to focus. So basically, we think that this transcription factor goes in and out of the nucleus much more rapidly compared to Mondo, but we don't know what this is really doing at this kind of dynamic localization or subcellular localization for this nucleus. But what is interesting about PTFB and C. elegans is that PTFB seems to affect the expression of many insulin-like peptides. So C. elegans has a lot of insulin-like peptides, roughly has about 40 of them. And they are taking over the function of many of the other peptide systems that we have seen in other models. So of the 40 different insulins, PTFB seems to modify the expression of the ones in blue, in the blue box. the ones in red modify the expression of PTFP. So it looks like there are things that PTFP is changing, and insulins can also change the PTFP expression, right? So the ones in blue have an E box in their promoter. So PTFP binds those promoters and then turns on transcription and gives you peptides from there, which you can then affect, right? So basically we said, let's test all of these peptides. Now you could ask, Shrek, why did you just look at insulins? Now, you're right. We should have done a full screen. C. elegans has 250 peptides. We shouldn't have just focused on insulins. But given the link with HLH30 or PTFB, we chose insulins for our current experiment. Now, there might be other signals that are coming from the intestine, and that's still something that we can pursue. And we want to look at other signals that the animal is using to relay this food deprived status. So I wouldn't be telling you all of this if there was no effect. One of these is INS31. INS31 is defective in response, no difference between well-fed and food deprived. Using the same strategy that we talked about earlier, if you take INS31 and you put it back just in the intestine, you start to get the difference back between well-fed and food deprived. If you put it in neurons, you don't get that difference. So it looks like INS31 is perhaps that signal that's acting downstream of Mondo and PTFP to relay the information of food deprivation from the intestine to the nucleus. Now, this is kind of an exciting observation that if these transcription factors are sitting in the cytoplasm, are affecting or inducing the intestine to release peptides, that might be a mechanism of how the intestine is communicating this information with other cells. And that's something that could be conserved in other places and maybe worth pursuing to test in other places as well. So it's an insulin. So what happens to the insulin, right? Insulin binds its receptor. And this receptor is DAF2 in C. elegans. So DAF2 C. elegans encodes one insulin-like receptor. It's a tyrosine kinase receptor. We have a mutation in DAF2 that we can test. These mutants in DAF2, the insulin receptor, have no difference between well-fed and food-deprived, not surprisingly, because they cannot tell that the intestine is sending them a signal. If you put the gene just back in all neurons, you get the difference back. You don't get it when you put it in intestine or in body wall muscle. So it looks like whatever signal is coming from the intestine is being sensed by the neurons to affect the behavior. Okay, so this is kind of where this paper stopped, and we proposed this model that DAF2 is working in neurons, insulin 31 from the insulin-like peptide 31 from the intestine relays that signal. And sorry, we published this paper and presented this particular model, right? So here's our current situation with this. In your food deprived, we think that HLH30 is causing expression of INS31. That INS31 then acts on neurons, acts on its receptor DAF2, which is on neurons, and then which gives you this behavior change between well-fed and food deprived. So food deprived animals are more likely to cross the barrier. Okay, so our paper ended here, but I have two additional bits of information that I want to share with you. This is unpublished data. We're still working on it, so it's kind of preliminary, and it's kind of hard for us to interpret this, which is why we kept it away from the paper, because we want to continue parsing out the actual mechanism of what is changing. Now, so what are the neurons that are receiving this peptide, right? I told you C. elegans has 302 neurons. So what are these different neurons, right? So those are the last two sections for our presentation this afternoon. So it turns out C. elegans has about 12 pairs of sensory neurons in the head that send dendrites to the nose, where they're sensing signals in the environment. And their cell bodies are in this ganglia, and their axons are where they connect with other neurons in the brain. Now, I told you that what happened with food deprivation is you're changing sensitivity to copper. Now, previous data had shown that there were three sets of neurons, three pairs of neurons, ASI, ASH, and ADL, which are known to detect copper stimuli specifically. Okay, so we are doing an experiment where we're asking, what are those three neurons doing? So before I do that, I want to show you what this plate looks like. So remember, we put a barrier of copper down the middle of the plate. Now there's an indicator that you can buy. This is a color indicator, which shows you how the copper is going to spread on this plate. So it turns out, and then in this assay, we typically put animals on one side and we have our odor that the animal likes on the other side. So if you look at this, you have a little gradient. The highest amount of copper is in the middle of the plate, and then you get some spread of copper from that midline across, which means as the animal is crossing this barrier, it's going to experience an increasing amount of copper until it gets to the middle area. And then after that, it starts to get a decreasing amount of copper. So the width of this copper barrier turns out to be about, 10 to 30 millimeters. Now, an animal is about a millimeter in length. So it has to cross a barrier that is almost 10 times or 30 times larger than its own body, which means as it's crossing this barrier, it's going to experience increasing amount of copper. Okay, so we try to replicate something like this in one of our assay systems. So here is our design. So a few years ago, we built this microfluidic device. So we take C. elegans, we stick it in the chip. It's a PDMS-based device. These are neurons that are expressing a genetically encoded calcium indicator. Outer channels control the flow. When you turn this one off, stimulus, which is normally going away from the animal, is coming towards the animal. So this allows us to stimulate the animal with a stimulus and then monitor the neural responses by changes in GCAM fluorescence in these neurons. Right? So the paradigm we used was this 1 second on, 1 second off stimulus. And we reason, our hypothesis is, if you go 1 second on, 1 second off, this might represent what the animal is experiencing as it crosses that barrier. Now, we are not able to change the concentration of copper in this device. So you're providing the animal with the same concentration of copper, which is not what the animal experiences. Remember, as you're crawling through the barrier, the animal is going to experience increasing amount of copper. So we can't do that, but we can give them more shots of copper over time. Now, we then monitor fluorescence. I'm going to show you a movie. There are two neurons that we are showing in this plane. This neuron is ASH, which is a chemosensory neuron. Here's the second neuron, ASI. So it turns out when you measure this, at 10 seconds, this movie is going to start hitting copper. ASH responds initially. You can see the flashes, 1 second on, 1 second off. ASI responds later. And then after a while, ASI then starts to become quiet. So ASI neurons have become adapted to the copper stimuli. So we think that's kind of what this response looks like. When you quantify it, this is kind of what happens. So in black is the data from well-fed animals responding to copper. And this is what you get. They start responding, maybe not the first pulse, maybe the third pulse. And then by about the seventh or eighth pulse, they're most active. And then after that, they start to adapt. Same neuron, if you food deprive them for 3 hours, they don't adapt. They keep responding to the copper stimuli. So we don't know what this means, which is why we didn't include it in the paper. But we get this increase in copper adaptation response. So the food deprived animal is not adapting, continues to respond to copper. Okay, so if you respond to copper, you become more active. So what does this mean? So we have one other thing that says that this is relevant. You could say, Shrek, you just showed us some data that is not relevant to this experiment or project. If you look at DAF2 animals, so DAF2 are these insulin receptor, tyrosine kinase receptor mutants. Those animals don't show that response. Well-fed animals are fine. They adapt, go back down. Food-deprived animals also seem to adapt and go back down. They don't continue to rise just like wild-type animals did. So there is some effect of ASI that's being changed in the DAF2 mutant, saying that perhaps this is somewhat relevant. So other data from our lab and from other labs has shown that ASI activity is suppressing turn behavior. So when you have in well-fed animals, you have less ASI activity because you're adapting. You get more turn. When you get turn, you're avoiding the copper barrier. In contrast, when you have more ASI activity, you don't have turns and you don't avoid the barrier, and so you cross the barrier. So this is kind of our crude model at the moment. More ASI activity means you cross the barrier, less ASI activity means you don't cross the barrier. So this is something that we have to prove either optogenetically or sonogenetically. Then we can kind of link ASI to the behavior, and we haven't been able to do that yet. This has been a very tricky neuron to do optogenetic experiments with. We haven't seen any effect of putting channelrhodopsin in these cells. Now, we don't know why that is, and we can talk about it if you guys are going to stay back in our discussion section. Okay, so broadly speaking, here is kind of where we are. So we are well-fed animals and food-deprived animals. Basically, when you're well-fed, you have mondo in the nucleus, PTFP in the cytoplasm. When you have food deprived, you now have mondo in the cytoplasm. PTFP is shuttling between nucleus and cytoplasm. These things are causing insulin peptides to be made or released. In well-fed, no insulin peptide is made or released. In food-deprived, insulin peptide is made and released. INS31 at least seems to act on tyrosine kinase receptor and maybe affecting some non-canonical signaling, which I didn't have time to show you. This is copper sensitive. This is copper insensitive. So this is kind of our model for how food deprivation is changing the behavior. Now, in the last 2 minutes, I want to share with you one other interesting bit of information, which I have absolutely no clue what it really means at the moment, right? So in my mind, I think C. elegans is an eternal optimist. You take C. elegans, put it on a piece of lawn. They once every hour, they leave this lawn and go explore the larger play. So this is C. elegans coming from bacteria, from the soil. They start entering the lawn from here. And then some of them stay here and some of them leave the lawn. Which means if you look at a population of animals, you're going to have populations in different amounts of food deprivation. Some might be well fed, and some might already be food deprived before you did your experiment. So we're asking the question, is there any communication going on between animals that are well fed and food deprived? So we're trying to differentiate within the populations of this stuff. Okay. So when you have well fed animals, they look like the one on the top. If you food deprive animals, when they're in the larval stage for 7 hours, they look like the one at the bottom. So they are different between each other. This is not the experiment that we are doing. This is a paper coming from Ryan Boss lab at Duke University. Our animals don't look this different between well-fed and food deprived. They look pretty similar because we are only doing 3 hours of food deprivation as an adult. Ryan is doing food deprivation as a juvenile during development. Now, it turns out C. elegans releases pheromones to communicate with each other. These pheromones have a synthesis pathway. They start their life as a fatty acid metabolite, and they are linked to the disaccharide ascarylose. And these pheromones are chemical based. They have fatty acid structures in it, and there are two enzymes, DAF22 and DHS28, that are required for the synthesis of these pheromones. Okay. So we did an experiment where we asked what happens in DAF22 mutants. DAF22 mutants, there is no difference between well-fed and food-deprived, saying that there is something that's happening in food-deprived animals with DAF22. So then we collaborated with Frank Schroeder's lab at Cornell University, and we asked what is the ascarocytes that are released from well-fed animals and compared them to the ascarocytes that are released from food-deprived animals. We just collected a whole bunch of these grew up a lot of cultures, collected the supernatant, sent it off to Frank. Frank does his thing, tells us what the ascarocytes are. Many ascaricides are not different. One ascaricide is mostly made in well-fed animals, not made in food-deprived animals. Okay, so we take that ascaricide and we add the ascaricide to the barrier in front of this copper barrier. Okay, so now when you do that, by itself, adding the thing doesn't change anything in well-fed animals, but it reduces the response in food-deprived animals. So perhaps ascaricides or the social cues are telling food-deprived animals that they are well-fed, and so they don't need to cross the barrier. Something, again, we are still trying to pursue and understand more in greater detail. But these are two aspects that are still outstanding, which is trying to understand what's happening in the neurons, and then trying to understand whether there is a social signal that communicates information about your food status to your friends. So if you are hungry, you're telling your friends that, hey, I'm hungry. And then if you are well fed, you're telling your friends, hey, we're all well fed. Maybe you individually are not well fed, but as a collective, maybe we are still well fed. And it's an interesting behavioral connotation for this, which we can talk about during our conversation. So what's the take home message? The take home is that we think intestinal cells are sensing internal food status, and they're sending that information to the chemosensory neurons. Mondo A and PTEF-B are two basic helix loop helix transcription factors that are likely acting together to relay this signal. Intestine is releasing INS31, which is probably acting on tyrosine kinase receptor DAF2 in neurons. We find that ASI neurons are changed in the response to copper, and that well-fed animals likely release this pheromone that affect the behavior of food-deprived animals. So there might be a social component going on. More broadly, we think that this idea that the intestine can tell neurons what's happening is not something that is unique to C. elegans. This has been proposed in mammals, right? I mean, leptin and ghrelin has been studied for many decades now. But what we are demonstrating in the worm is the underlying mechanism, the role of these transcription factors in driving this process or in initiating the process is interesting and perhaps can be tested in other places here. So I didn't have time to talk about other projects in the lab. I have 5 postdocs, 2 grad students, research scientists, a couple of research assistants, and undergrads who work with us. We have a number of collaborators who are working with us on different projects. And here is everybody in our lab and our funding sources. And I'm happy to take questions. Obviously, I can also stay later if you have more things to discuss. Thank you so much for your attention and looking forward to question time.
Okay. So thank you so much for this great talk. I really enjoyed it, and I can see that we have some questions from the audience. So we can start with a question from Ibrahim. It says, cool work. How is food deprivation sensed and translated into ML1 translocation? Maybe I missed it in the talk. Is it circulating GCP levels or
Ibrahim,
something
that's
else?
a really good question. The honest answer is we don't know. We think that G6P is the actual molecule that affects the Mondo translocation or MML1 translocation. However, we haven't shown that directly. We haven't shown that G6P is changing in the intestine. We are assuming it is changing because as the animal is food deprived, there is less glucose and then so less G6P. But we haven't demonstrated that directly. Rita is asking a second question. Rita is, go ahead, sorry.
Yes. No problem. So really interesting talk. Could this behavior be dependent on the visual system of the animal? I was wondering how the food quality could also influence risk-taking decisions.
Yes, so Rita is asking another question about the visual system. So C. elegans, to a large extent, is blind. It cannot see very well. It can see blue light. We think blue light represents polarized light that the animal can experience early in the morning, maybe stays away from the surface of the soil and stays inside the soil. But we don't think it sees any information. It sees anything like that. Now, obviously, in different systems, in a different model, you could have multiple different inputs coming in. So when you study flies or fish or mice, you might have different sensory systems that are changing. And this is a really interesting question in these models, right? So for example, if you food deprive an animal, what sensory modalities are actually changing? What sensory modalities are changing can tell you something about what this animal has evolved to do in its natural environment. C. elegans lives in topsoil, which means that it's living in the top 1 or 2 inches of soil, mostly in moist climates, in rotting fruit and rotting bacteria, rotting vegetables, eating bacteria, interacting with other nematodes and stuff in its environment. So for this chemical signal, mechanical signal, proprioception, osmotic shock, these seem to be the sensory modalities that C. elegans cares about. Of course, pheromones, because they are a lot of nematodes in the soil. And then apart from pheromones, temperature changes is very important for a worm. And for a worm, moisture content is very important because it cannot move if the soil is too dry. And so those are the things that the animal really cares about. If I was studying flies or fly larvae, or if I was studying a mouse, I would look at other modalities and ask what are the different changes there. Uh, we haven't done exhaustive search, and you're right about that. So your point is valid that have I tested temperature? Uh, we don't know that. We have only tested all chemosensory behaviors, but we can look at other modalities like pheromone sensing ability or temperature sensing, and that's something that we should do and we haven't done yet. Because food deprivation might have effects on other systems as well.
Great, thank you for that. Great answer. Then we have from Sita. Very interesting. Do you have any hint on which neurons could be mediating this brain-gut communication? Do you think it is longer distance communication or local intervention
Yeah,
of
so
the gut?
Zita, this is a quirk of C. elegans. So in C. elegans, all of the body organs, including the neurons in the intestine, are basically floating in a coelomic fluid. And so if the intestine releases something into the fluid, that whatever that something is, if it's a metabolite or a peptide, it is transported all across the animal. And so every cell can have access to that. Now, obviously, that's not the case in a more complex animal because there are circulatory pathways, blood circulation and lymphatic circulation that carry signals from one structure to another. But this is something that is that's a quirk of C. elegans. Now, I showed you one-way information flow from the intestine to the neurons. But you're right, the neurons can also talk to the intestine. And we have a second story that's currently in review at Nature Aging, showing that neurons are releasing a peptide that affects the function of the intestine in the context of aging. So it looks like there is bidirectional communication. It's not just the gut talking to the neurons, but also the neurons can talk to the gut. And in this case, it looks like the communication is long distance by peptides. Now, there are 8 neurons in C. elegans that are very close to the intestine. But as far as we can tell, we don't know what these neurons are doing. Um, you know, the other secret is that even though there are 30 neurons in C. elegans, 302 neurons in C. elegans, everybody studies like 30 of them. So the rest of the neurons, nobody really studies. They're not as good reagents for that. But we have no idea whether these other neurons are directly affecting the gut. One possibility is that the neurons that are close to the intestine are really affecting the motility of the food, because the intestine has to contract and expand to push the food from one part of the intestine to the other. So that's probably what those neurons are doing, although this is all speculation. We have no reason to think that, because we find that these long distance communication are happening not from those cells but from other places.
Great. And then we have one last question from the audience, from Ibrahim as well. Your behavioral experiments show that with high copper concentration, the mutants still avoid. Do you use the same concentration in ASI imaging experiments and see the mutants adapt at high concentrations?
Yeah, Ibrahim, this is a really good question. I wish you were in my lab. We have not done that experiment. In our behavior experiments, we have done 25 to 50 millimolar concentration of copper, and we have only tested 10 to 25 millimolar in the imaging device. The problem is, I think there is a little bit of a larger problem. So in behavior, we have put this line of repellent, and then the repellent has absorbed onto the agar surface and started to spread. Now, what the animal is actually experiencing might be very different in that behavior assay versus what we are doing in our imaging device. In our imaging device, the animal is stuck and you're spraying a bunch of chemical on its nose. So this might be much higher than what the animal experiences on the agar. Or it could be much lower. I mean, we don't, depending on how much is getting past the cuticle of the animal. So there is still a bunch of unknown questions about the neuronal responses. And I think the neurons are fascinating. It's very interesting to study, but we don't. So we kind of had to cut our paper a little bit. I mean, I would have wanted to include the neurons and the social signaling part of it, but our postdocs were applying for jobs and we needed to publish our work, so we kind of reduced the emphasis so that we can get our paper out. But the neurons are definitely an interesting angle to look at, and we only looked at 3 of them, and there might be other neurons that we want to see. And we now have the ability to look at animals look at all the neurons in the head of the animal, not just in trapped animals, but also in freely moving animals. So we might be able to get a broader view of what is changing in a food deprived animal. And remember, we talk about food deprivation as 1 hour, 2 hour, 3 hours, right? This might have no meaning for C. elegans. For C. elegans, number of hours doesn't mean anything, because food deprivation is a continuum of responses. And we might have to get an idea by defining particular time points when there are changes in the nervous system that might correlate with behavioral changes. So volumetric imaging is happening. We do freely moving animals now, so maybe we can get an idea of what's going on here.
So now we are moving to Zoom for some more discussion. You can see the link in the chat. So if you have more questions for Dr. Chalasani, you can follow us there. Otherwise, I just want to thank you again for this great talk. It was so nice to listen to it. And I also want to say want to thank the audience, and I want to say that next week we will have Jeremy Borninger, who will tell us about identifying central mechanism of glucocorticoid circadian rhythm dysfunction in breast cancer. So that is the topic for our seminar next week. So now we're moving to Zoom. So thank
Meet you all in Zoom
everyone.
in like